Um, we'll go ahead and start today's business meeting for the North Carolina Fossil Club. We're delighted today to welcome Dr. Bobby Bossenecker from the College of Charleston. Bobby hardly needs an introduction. He has a really big reputation amongst the members of the North Carolina Fossil Club based on the extent of collaboration that exists between himself and members of the club, who seem to be extremely delighted to find out that they have found a fossil that will be of interest to him. So I know everyone's really keen to hear today the results of your latest research on these fossils that are familiar to us. So please, we are really looking forward to it. And thank you very much for being with us here today.
Absolutely. It's my pleasure to speak with you guys today.
Bobby, you've ended up muted again.
Oh, sorry. Our point of clarification: are you guys doing the normal business meeting first, or are we going straight into the talk?
We're going straight into the talk so that you don't have to listen to our business meeting.
All right. Not that I wouldn't mind that, but here we go. So my wife Sarah and I moved to Charleston in 2015, and we got invited to go to our first Aurora Fossil Festival in 2016. And one of our first things we did was, go to Belgrade Quarry, and we've been back several times, both trips organized, I think, through the former Special Friends of the Aurora Fossil Museum, and then other trips organized by North Carolina Fossil Club. And we found a few things ourselves, but most of the really important stuff have been found mostly by members of your club. So I think it was 2017 that I gave a talk on fossils from the Lee Creek Mine and Charleston, South Carolina, and comparing and contrasting the two, Oligocene fossils and Miocene fossils. Well, the interesting thing about Belgrade is that it seems to be in the middle. So now that we've amassed a bit of a collection from Belgrade, I can come back and talk about what you guys have found and how it differs from Charleston or from the Pungo River limestone at the Lee Creek Mine. All right, fossil cetaceans have a pretty amazing fossil record. They're pretty famous for the land to sea transition, and so we've got a couple of very early cetaceans like Georgocetus on the bottom, but this is mostly a cladogram highlighting relationships within Neoceti, the baleen whale, toothed whale clade. We've got baleen whales on the bottom, mysticetes are the technical name of baleen whales, and then the odontocetes are the toothed whales. I prefer the term echolocating whales because early baleen whales have teeth, as do the archaeocetes. And not all toothed whales have teeth. Teeth have been lost like a half dozen times in different groups. Think like a narwhal, they just have a single tusk. Beaked whales as well, and females of both groups don't have any adult teeth that are erupted anyway. The earliest fossils of cetaceans are, of course, archaeocetes. They show up in the early Eocene in India and Pakistan, and by the middle Eocene are basically worldwide. By the time you get into the late Eocene, we get basilosaurid whales like Zygorrhiza, Basilosaurus, Syntheocetus, etc. Fully marine, vestigial hind legs that may have functioned as copulatory grips, but not for locomotion at all. The Oligocene epoch is what I study the most here in Charleston. And we've got the early diversification of both baleen whales and echolocating whales during the Oligocene. So in Charleston, we've got two rock units, the Ashley and the Chandler Bridge formations, which are a really unique window into this time period because there were generally low sea levels during the Oligocene. So there's not a whole lot of, in terms of volume of marine rocks with fossil cetaceans from the Oligocene anywhere, which also explains why I went to New Zealand for my PhD. New Zealand and South Carolina probably have 3/4 of the world's fossil cetaceans from the Oligocene epoch. The remaining quarter are from places like Japan, one fossil site in Australia, the Pacific Northwest, Baja California, and a few scattered sites in Eastern Europe and the Caucasus. The Miocene, on the other hand, is really well represented. We have relatively high sea levels throughout the Miocene. And we've got some of the most spectacular fossil cetacean assemblages in the world from places like Calvert Cliffs, and fragmentary but decent collections from the Lee Creek mine, from the Punga River limestone, which are largely tooth and ear bone based fossil assemblages, especially for the Miocene part, which fortunately is very comparable with the Belgrade Formation. So in the Miocene, we get the first modern families appear. So the earliest members of the humpback whale group, the earliest oceanic dolphins, porpoises, belugas, etc. And there's a host of extinct groups and some stragglers from the Oligocene that make it into the early Miocene. You get into the Pliocene, we have the first records of extant genera. So you have a lot of extinct species that belong to extant genera. So for example, at the Lee Creek mine, there is Lagonorhynchus harmatucki, named after Pete Harmatuck. Lagonorhynchus is the modern genus of white-sided dolphin, but it's an extinct species. And then by the time we get into the Pleistocene, we get mostly extant species with a few examples of extinct species, largely in modern genera. The Pliocene, we do have a fair number of extinct genera and species. So the Pliocene is kind of a neat mix. And the two time periods I really like to focus on are the Pliocene and the Oligocene. The Miocene I've generally found it's overdone, and because there's a lot of fossils from it, and there are more interesting questions to be had at the beginning and sort of the last 5 million years of Neocene evolution. So we'll be talking about the Oligocene for the rest of this, and to a degree the early Miocene. All right, so Earth over the last 65 million years, there are a few major paleogeographic events and climatic events. So during the Middle Eocene, this is basically the last period of time where we do not have a circumpolar current. So Australia and South America are still relatively close to Antarctica. Remember, they are parts of the landmass Gondwana, the southern continents. And at the end of the Eocene, they pull apart, allowing this circumpolar current to form, which basically leads to runaway development of ice sheets. So there's this big cooling event across the Eocene-Oligocene boundary. We'll talk about the biotic effects here in a minute. But one of these abiotic events is the formation of the Antarctic ice sheet that stays there for the next 35 million years. During the Eocene, we've also got the collision of India and Asia, forming the uplift of the Himalaya, which resulted in the Earth's third ice cap right at the equator, or close enough to it, which reflects a little bit of light and probably led to some of this cooling as well. There is another cooling event, and it's hard to see here, but if you look at the red line, there's a little dip in it right at the Oligocene-Miocene boundary. It's not as severe as the event at the Eocene-Oligocene boundary, but it is faster. And some changes in the cetacean fauna across the Oligocene-Miocene boundary have been tied to that short-term glacial event. In the late Miocene, we get continued cooling, and then runaway cooling in the Pliocene leading up to the Ice Age during the Pleistocene. So if we look at marine faunas through time, generally speaking, in the mid-Cenozoic, we start off with relatively low diversity cetacean assemblages, mostly dominated by basilar sword whales, a large number of sea turtles, relatively small megatooth sharks, and sort of a low diversity of pelagic fish, but a lot of fish groups show up for the first time during the late Eocene, especially all of the members of the tetraodontiforms, ocean sunfish, pufferfish, porcupine fish, triggerfish, etc. A lot of those all trace their origins back to the Eocene. During the Oligocene, we still have multi-species assemblages of sea turtles, so higher diversity of sea turtle than we have today, slightly bigger megatooth sharks, a more diverse pelagic fish fauna, but an explosion in cetacean diversity. So we've got several toothless baleen whales, several tooth-bearing baleen whales, and just a bucket load of echolocating whales, including little tiny dwarfodonticetes, and some of the first big macro-predatory dolphins. When we get into the Miocene, we have very, very few, at least we're talking about the earliest Miocene here, very, very few baleen whales, along with the first squalodonids, a lower diversity of small dolphins, but we get the first swordfish dolphins up there on the upper left, which are the long-snouted Urinodelphinids. Sea turtles are much less diverse. We also get the first sperm whales and fewer pelagic fish. And I'm basing the pelagic fish here mostly on the record of swordfish. And of course, we get the first truly gigantic megatooth sharks like Carcharicles chipatensis, and eventually Megalodon during the Middle Miocene. So to put that into the context of the slide before that, we get this radiation of cetaceans driven by this climate crash, which you normally don't think of in terms of evolution. Normally, land mammals tend to diversify during warm periods, but cold periods, usually in the ocean, end up with a proliferation in diversity and numerical abundance of plankton, leading to diversification of many marine faunal groups. So we got a pretty high billfish diversity, a bunch of mackerel shark groups that go extinct across the Eocene-Oligocene boundary, radiation of reef sharks, the giant sea snakes, which you may or may not have even known about during the Eocene, those go extinct at the Eocene-Oligocene boundary. And then, of course, within cetaceans, we have this diversification in feeding morphology and hunting. So filter feeding evolves for the first time, as does echolocation. A lot of these early lineages last less than 10 million years and go extinct at the either at the end of the Oligocene or during the Miocene. Sorry, that should say end of Oligocene. There's actually this thing for the first 5 million years of the Miocene called the baleen whale gap, where globally you generally don't find any baleen whale fossils. And for example, even in the late early Miocene, Punga River limestone, there are virtually 0 baleen whales. Out of the Rita McDaniel collection that was donated to us a few years back, we had 3 confirmed cetacean ear bones or baleen whale ear bones from the Pungo, as opposed to literally, several hundred odontocete ear bones that she had found. Leatherback sea turtles and billfish dwindle and diversify, but we get chipatensis and Megalodon. And then shortly thereafter, when it's really warm in the Middle Miocene, we get the earliest records of modern families, and shark and fish faunas become basically modernized about 10 to 15 million years before cetacean fossil assemblages become modernized. And then we get, those modern marine mammal and sea turtle genera during the Pliocene and Pleistocene. All right, so with all that global stuff out of the way, now we can bring it back locally. I'm going to talk first about Charleston because the fossils from Charleston are a lot more complete and serve as an important benchmark for comparison with Belgrade. Folks started finding shark teeth and other fossils really early in Charleston. This is from Agassiz monograph in the 1840s. Often, big ass scary shark teeth are probably going to be one of the first notable fossils anybody talks about from a new area. During the Ashley phosphate mining days in the 1870s and 80s, loads of cetacean fossils and other marine mammal fossils were found. So we got a bunch of Ziphiid skulls here, beaked whales. There's a walrus tusk, admittedly Pliocene, there in the middle, and some sperm whale teeth. And this is just a small sampling of fossils that Joseph Leidy published in 1877. Granted, without any locality data, we assume some of this is dredged. For example, some of these specimens that when you look at them in person, have oysters attached to them. Others were probably dug up from the Ashley phosphate beds themselves, which are on land deposits at the base of the Pleistocene. So it's everything reworked from the Oligocene, Miocene, and Pliocene into the basal Pleistocene that they strip mined. And one thing that happened after the end of the Civil War, you have this large freed population that needs work, and these plantations that own a lot of land suddenly are aware that they have phosphate underground, and they can't really afford to pay all these former slaves to come farm the land anymore. So what they started doing is leasing out the land for phosphate mining. And so if you go into the plantations along the Ashley River, only a few miles away from where I'm talking right now, you'll see these big rows of earth that are about 3 meters tall, separated by a ditch, and then each ridge will go for a couple hundred yards, and they'll be spaced apart about 10 yards apart. And you just see this on Highway 61 driving along the south side of the Ashley River, ridge after ridge after ridge, with about 100 to 140-year-old trees on top. None of these trees are old growth. They're all maybe only about 1 to 2 feet in diameter. That's all post-Ashley phosphate mining trees. And once you get close to the plantations themselves, those ridges stop, and you have really old growth trees, including like in the rows along the carriage road up to the house. These mines were frequently dug out by recently freed slaves, and many contemporary journalists observed that the safety and conditions were not much better than before the Civil War. Here's an example of how it worked. At first, it was just with shovels and buckets and wheelbarrows, but eventually they started putting in little minecart tracks. They would use oxen and plows to dig away a lot of the topsoil. You take all the phosphate and dump it into these chutes and literally dissolve it. Well, first you'd wash all the sand and gravel off of it that was not phosphatic. Then you'd take all the nodules and they'd literally dissolve it in acid. And then you'd end up at the end with phosphoric acid in barrels that would be shipped off for agricultural fertilizer. The map on the lower left shows the Charleston area. Downtown is to the lower right. We've got the Ashley River and the Cooper River on either side. And all the phosphate mining is generally actually right around where all the plantations are, and that sort of pink salmon color. All of that was dug by hand. That is dozens of square miles of earth that was tilled by hand and mined and processed for phosphate at these plants along the rivers. The gray, that's artificial fill, so that's more recent. The artificial fill still contains all the phosphate. So Drum Island, right underneath the Cooper River Bridge, is a popular spot to go looking. And literally they dredge out the deeper channels in the rivers and then dump all that artificial fill into anywhere where it's gray like that. And in some cases, like downtown Charleston built up on it. Although in downtown Charleston, it's actually mostly like garbage and stuff from the early 19th century that the really expensive homes down in the Battery are built upon. They're built on trash. On the lower right is a look actually in a screen of mine from last winter after I did some digging in a pristine, unmined exposure of the Ashley phosphate beds, only about a mile away from my house. So the danger of living up in North Charleston and Somerville is that whenever there's a new construction site, it means that there's a very richly fossiliferous fossil site within a 2-minute drive of my house, which means you might end up eating dinner or about to eat dinner and thinking, well, I got another hour of sunlight. I can roll the dice and see if I can find a megalodon tooth or something more interesting. Oh, forgot to click that. That's basically the Ashley Phosphate Mining District there. And a lot of the really old famous plantations like Drayton Hall, Magnolia, Middleton Place, Runnymede, those are all sort of towards the upper left part of that where that big meander is. All right, so one problem with the Ashley Phosphate mining days was that nobody knew much in the way of geology. There were some observations from before the Civil War, including stratigraphic work by Charles Lyell, of all people, who spent several months, if not almost a year or so, in the American Southeast. And so Lyell named the Cooper Group, if memory serves, or the Cooper Marl, which in those days was thought to be entirely Eocene and may have even actually very likely named before the Oligocene as a time period was even conceptualized. A lot of construction sites and USGS mapping during the 1960s and 70s really revealed vertical access to the stratigraphy for the first time since the 19th century. Paleontologists went out to these construction sites and dug up lots of fossil cetaceans, at least from the Oligocene parts, and then a series of limestone quarries put in the, I think, 1960s near Harleyville, like Giant Quarry, Blue Circle, et cetera, revealed all of these much earlier cetaceans from the Eocene. So from the cross member of the Tupelo Bay Formation, formerly the Cooper Marl, we have Carolinocetus gingerichi, which is a 4-legged protocetid whale, the same group as some of the specimens from the Castle Hayne that David Sanderson's been collecting recently. And then from the pregnal member of the Tupelo Bay, as well as the Harleyville and the Parker's Ferry Formation, we have a small fauna of basilosaur whales, which are still admittedly 4-legged, but their hind legs are vestigial. And you basically have the same sequence of limestone in North Carolina. It's just the Castle Hayne up there. Slightly different suite of fossils. Instead of Carolinocetus, y'all have Crenatocetus. Yeah, I can't remember the species name off of it, but that's a protocetid named by Sam McLeod and Larry Barnes about 10 years ago. In the Ashley and the Chandler Bridge Formation, we've got a lot of early echolocating dolphins and early baleen whales. And then there's a big time gap. We don't have much in the way of Miocene here. People do find occasional Paracaricles chibatensis teeth, and I've been collecting a few basically spoil sites where everything's ex situ, but we do have a lot of the same dolphin ear bones that you find in the Punga River limestone, but we don't have the rocks. We think it's the Mark's Head Formation, which is a lateral equivalent of the Pungo that is better exposed around Savannah, Georgia. Oh, and before I continue, the Ashley Formation is about 28 to 30 million years old, so early Oligocene, and the Chandler Bridge is late Oligocene, roughly 25 to 23 million. So, and there are slightly different faunas between the Ashley and the Chandler Bridge. The differences are not enormous, though. The faunas are quite similar, and there's differences on a species and occasionally a genus level, but you have many genera in both, and at least many families in both, and some species in both rock units. Some of the earliest fossils are right here. So Agarothius pygmaeus was discovered somewhere out of Middleton Place plantation out in West Ashley from the Ashley Formation and borrowed by Agassiz right before the Civil War. And the skull was lost during the Civil War. Nobody knows where it is. My PhD advisor, Ewan Fordyce, discovered this tooth, which is the missing tooth that must have been snapped off of the skull at some point. And he found that, I think in Harvard Museum of Comparative Zoology, even though the specimen belongs to Academy of Natural Sciences in Philadelphia. I might have that backwards. It was definitely at the other institution, just sitting in a drawer unaccompanied, probably sitting there since the 1890s. On the upper right, we've got Diapotherium manigoi. That is derived from the Ashley Formation, and that was dredged off of the bottom of the Wando River at the very beginning of phosphate mining. One of the first modern discoveries, and I use that term loosely, and by modern, I mean the specimen has basically been in the same collection since it was found, is Xenophis sloanii. It was collected at around 1900, named by Remington Kellogg in the 1920s. I think 20s. I always get that mixed up. And it's this, not an enormous skull. It's an animal about the size of a common dolphin, but it's got these really enormous cheek teeth. I'll talk more about xenoraphids here shortly. Unlike Agarothius, we still have the holotype specimen of Xenophis sloanii, and that's actually a picture I took. Which reminds me, I really need to get Mace Brown Museum scale bars, because Victor Perez and others always comment on me always using their scale bars. They love it because it's free advertising. But anyway, more significant discoveries started being made in the 1960s and 70s, chiefly by Al Sanders, who just passed away about a year and a half ago, sadly. This is a photo I took of Al Sanders in his screened-in porch out on Folly Beach with my PhD advisor, Ewan Fordyce there, who himself just retired about a month ago. Some of Ewan's postdoc research was on fossils from Charleston, but Al's the one who went out and dug up a lot of these. So one of them is this beautiful skeleton on the upper left, which is he didn't name. No, he helped name, along with Bruce Erickson, Gavialosuchus carolinensis, which is like a 30-foot long, or at least 20 to 25-foot long estuarine crocodile, closely related to the modern false gharial Tomistoma, I think. I don't know a whole lot about crocodilians, but it is a pretty spectacular fossil. That skull's like 4.5, 5 feet long. And then on the bottom is this dolphin that Al found in the early 1970s and nicknamed Genus Y. And his name has stuck around for a really long time. The problem is Al started digging up fossil whales and dolphins from only about 3 miles away from here at the type locality of the Chandler Bridge Formation, where they dug out, they did a gridded excavation in the Eagle Creek subdivision. And I think it was like, I don't know, like 10 by 30 yards, something like that. And out of that came like a dozen dolphin skeletons. And I think this was one of them. And so we got just this influx of beautiful dolphin fossils all at the same time, and he was just inundated with too many fossils to describe. The Charleston Museum collection, our collection at Mace Brown Museum is pretty nice, but it pales in comparison to Charleston Museum. They're at least 2 to 3 times the size of our collection, and the local material, I mean. And so Al started nicknaming them Genus X, Genus Y, Genus Z, Genus A, B, C, all the way up to G, H, and I. Some of them might be conspecific. Al is a little bit more of a splitter than I am. But anyway, last summer we named this. Oh, wait, I'll come back to Ankylorhiza. Sorry. Now, Ewan's retired and mostly worked on New Zealand fossils after his postdoc, and Al Sanders passed away. But Charleston paleontology is very much thriving right now. I can't take master's or PhD students, which is fine because I'm not paid enough for that anyway. I do love taking undergraduate projects on because they tend to last less than a year. They're nice and manageable. And there are all these projects I would like to do, but I don't really have time to. So I can train a up and coming paleontologist to do the research themselves. So one of these that made the news in November is the first nursery assemblage for Carcharocles angustidens, worked on by my student Addie Miller. It has survived the first round of peer review. We tried to make the title include the Baby Shark song lyrics, but the reviewers were a bunch of boring sticks in the mud and told us, I don't get it. So we had to change it, but that's all right. The paper's coming. Addie's working diligently on the revisions right now. One of the more important sites that Sarah and I worked on, and that's actually Sarah there taking a picture of the sunset, is this locality. Again, this is only like 3 miles away from our house. We had access to the site for about 6-7 years. One of the best exposures of the Oligocene sequence anywhere I've seen in Charleston, and probably since the 1980s, based on discussions I've had. We found this bird skeleton in there, which turns out to be from booby or early gannet, some kind of sulid bird. And we've got much of both wings, part of the skull, about half the vertebral column, parts of the limbs, etc. And this is one of the oldest known members of the Sulidae, and probably the most complete sulid skeleton known before the late Miocene. And, just adorably wonderful birds. So that's a project I'm working on with a colleague at Clemson, Adam Smith. One of my students, Bailey Fallon, published on a series of leatherback sea turtle fossils just a few months ago. Here's Sarah with a unique radial ridged specimen of Staphophorus. There's also some much more robust leatherbacks that have ossicles that are almost an inch thick. Modern leatherbacks have shells that are 1/4 inch to 1/8 of an inch thick. So there's some real weirdness going on with these early assemblages. The Oligocene of Charleston has 3 species of leatherbacks all at the same time. There's only 1 modern species of leatherback, and the Oligocene is basically the last time we had more than 1 species in the same rock unit. So ecologically, there's something interesting happening. We're not really sure what, though. A fair number of fossil sea cows, actually the most diverse sea cow assemblage in the world. So again, reflecting something ecologically interesting. Jorge Velez-Jarbe published some of his PhD research talking about niche partitioning, and basically these sea cows were all eating different types of food. That was when the Oligocene assemblage was only known to have 3 species. Well, now it's got 6. So the Oligocene is a really unusual period of time. And I've only got 4 of them up here. There's 2 more that I can't even fit on this slide. All right, finally the whales. Charleston's got a lot of very, very important whales. Some of the best examples of Oligocene baleen whales and dolphins anywhere on Earth. One of the most important ones to me is this, Eomysticetus whitmeri. There are much better specimens now. I named 5 species of Eomystacetids from New Zealand coming out of my PhD. And I had to compare all of them with this fossil found in the early 1970s by Al Sanders. I call these surfboard whales because the rostrum or snout is about the shape of a surfboard, and they're about 6 feet long. So just the size of a relatively short board. I mentioned the giant dolphin genus Y earlier. We recently named this Ankylorhiza tiebmani. The original specimen in Squalodon tiebmani was just a chunk of a snout dredged off the bottom of the Cooper River, or the Wanda River rather, in the 1880s. And one of the new specimens is this beautifully complete skeleton, basically missing the lower jaw and some of the vertebrae and some of the distal parts of the flipper. But it's got a meter-long skull, so it's almost the size of Squalodon whitmeri, the giant Squalodon from the Calvert, and a lot bigger than Squalodon calvertensis. It is not a squalodonid, though. It is superficially evolved into a squalodon-like skull shape. But the top of the skull looks very, very different from Squalodon. So squalodontids as a body form have sort of evolved multiple times, which is kind of unusual. The most diverse and probably the most important group of dolphins are the xenorhophids, at least from Charleston. And there's a lot of them. Xenorhophis slonii, I already showed you there. We've got more specimens of Xenorhophis slonii, which I used to think were a different species. Turns out I thought so because they're all adults, and slonii looks weird because it's a baby. So that's what I'm working on right now. In 2017, we named Inermerostrum xenops, which is that tiny little skull there, which happens to be the smallest dolphin ever. No teeth, short snout. We think it was a dwarf toothless suction-feeding dolphin that maybe got to about 4 feet long at the largest. Also small, but very toothless, or sorry, very toothy, are Echovenator sandersi, which I'll talk more about in a moment, Albertocetus mephrodorum, and then slightly bigger is Cotylocara masai, and then this new genus on the lower right over at Charleston Museum. That's actually from Al Sanders's original Chandler Bridge excavation. All right. So that's the background here in Charleston. So let's move on to the Belgrade Formation. There have been some really confusing differences in the terminology used for Oligocene rock units in southeastern North Carolina. Some folks have called the Belgrade and the River Bend the same thing, or the Belgrade have included the upper part of the River Bend into the Belgrade, and it's just kind of unfortunate, the degree of stratigraphic confusion. But at present, at a Belgrade quarry, we've got the Castle Hayne on the bottom, the 2 different members of the River Bend, and then a relatively thin exposure of the Belgrade Formation on top of that. And then Pliopleistocene rocks like the Waccamaw, above, or maybe the Duplin. I think it's the Duplin actually, above at the very top. As Bruce has talked about at length, the land mammals suggest a late Araucarian stage assignment, so AR4, and that suggests an age of about 18 to 23 million, so early Miocene in general. Work on the mollusks suggests a late Oligocene age exclusively. Microfossils, on the other hand, reflect deposition on either side of the boundary. So around 27 to 23 million, just eking into the Miocene a little bit. Strontium isotopes from the base of the Belgrade indicate a maximum age of about 26 million. So unlike mollusks and land mammals, I think the best evidence comes from strontium and the microfossils, indicating that the Belgrade spans the Oligocene-Miocene boundary. Which is really interesting because it's one of the only fossil sites in the world where you actually get fossil marine mammals from right about the Oligocene boundary itself preserved. One of the only other spots that comes to mind is unfortunately named Horace Quarry, but it's spelled H-A-U-G-H. It's a weird Irish word, not a W, and that's in New Zealand. And I published a few eomystacetid whales from, we usually try and call it Hackateramia Quarry, which is the Māori name for it. And that also covers the boundary. There are no similarities between the New Zealand Oligocene boundary locality and the Belgrade, aside from sharing eomystacetid whales in general. But aside from that, I can't really think of any other fossil sites that span the boundary. Maybe the Nye Mudstone in Oregon, but there's not a whole lot known from that. And the base of the Clallam Formation in Washington, but again, not a whole lot known. A couple skulls, but not a fauna like is known from Belgrade. The Castle Hayne has, as I mentioned, basilosaurids like Basilotritus and Cynthiacetus. Basilotritus used to be Eocetus wardii, originally interpreted by Mark Ewan as a big protocetid, now known to just be a really weird early basilosaurid. We got protocetids like Cronatocetus, and then these things that I call furry otter crocodiles, the Remingtonocetids, just reported about a month ago by Mark Ewan. From the Belgrade, we've got an unusual assemblage, including sperm whales, Uriah delphinids, squall delphinids, Whipatiids, agarophiids, eomystacetid whales, xenoropid dolphins, and then interestingly enough, squalodon and Ankyla rhiza. And the Castle Hayne cetaceans are largely known from other localities, but all of these Belgrade specimens up here are all from the quarry. And there's a few other specimens at the Smithsonian that are from Onslow Beach and North Topsail, but I'm not, because I can't go and study those specimens right now because of COVID, I'm going to submit the paper just based on the Belgrade fossils collected by you guys and Sarah and I. All right, before I continue, a bit of a warning here. The rest of this is going to really focus a lot on ear bones. Ear bones are super weird, and they're sort of, unlike shark teeth, they're sort of a real acquired taste. Most people don't like them, or at best, they're indifferent to them. They're like, I don't really know what it is. It looks weird. It's got some holes in it. It looks kind of like a piece of popcorn or a potato, depending upon what element it is. Which is convenient for me. If people don't like them, it means they're much more amenable to coughing them up and helping me out with my research. So we've got a baleen whale on the bottom and a dolphin at the top. And I think that's a 2-centimeter scale bar in the white boxes. So obviously baleen whales have really big ear bones, dolphins have small ear bones, but there have been small baleen whales in the past and large dolphins in the past. So the two styles of tiny and big ear bones, there's more of an overlap in the past. These bones are formerly part of the squamosal, which is the bone right here in your skull that your jaw attaches to and the back of your zygomatic arch articulates with. These are having the ear bone separated from the skull is basically an adaptation for reducing bone-conducted sounds. Sound waves travel really fast through water, and there's not a significant enough change in density between water, flesh, and bone for the sound waves to bounce at all. So it basically just each sound wave will arrive at left and right ear at the same time, which is why you cannot hear directionally underwater in a pool. Cetaceans, on the other hand, they separate the ear bones out from the squamosal and the ear bones from each other and surround them with air sinuses, and then have this little fat tube, literally a tube of acoustic fat, which is that little pink cone that focuses in the sound waves just like your ear canal, only it's internal and it's a difference in density between the air and the acoustic fat. Here's a cross-section of a bottlenose dolphin middle ear. So at least just the bone. So we've got the tympanic bulla, most of it's cut away here on the left. And we got the periotic. Periotic is the same thing as the petrosal. And then in red is the malleus, otherwise known as the hammer. In yellow is the incus, known as the anvil. And then green is the stapes, the stirrup. And the way this works right now, what you're hearing, sound waves, and you're cheating a little bit if you're listening to headphones that have stereo, but if you're not, the sound waves arrive at left and right ears, get funneled into your external ear canal, bounce off the tympanic membrane, which is like a drum. That drum basically hits the malleus, which is dense, but not as small and not as dense as the incus. So the sound waves change frequency when they get to each smaller ear bone. So malleus hits the incus, hammer hits the anvil, anvil hits the stirrup, the stapes, and the stapes goes in and out of this opening in the, which is basically the opening to the cochlea. Your cochlea is filled with cochlear fluid. That fluid makes these little waves that go up the spiral, and each hair there, and yeah, the cochlea is filled with hair, which now that I say it out loud, it sounds kind of gross. But those hairs, each hair detects a single narrow band of frequency. And the higher up the coil you go, the higher the frequency of the sounds. Each hair has its own nerve, and those nerves coalesce and form basically like a huge fiber optic bundle that goes straight into your brain, and your brain takes those different signals and converts into the sound you're hearing right now. All of this is happening right this moment. In a whale, there's no external ear. There's no eardrum anymore. The tympanic bulla basically sort of functions as the eardrum. So you end up with sound waves going through this acoustic fat cone, hit the ear, the tympanic bulla, and the tympanic bulla has this huge dense part, which is on the right-hand side, the lower left thickened part, which is the part that usually fossilizes. The rest of it usually breaks away. That vibrates, and it hangs off the bottom of the skull, connected to the periotic only through the malleus. So I liken it to an acoustic pendulum. The pendulum swings around, all those vibrations are funneled into the malleus. And then the rest of the process is the same. So the mammalian ear, and remember, the tympanic bulla, malleus, incus, and stapes all used to be bones in the jaw joint of reptiles. So they've all been co-opted for this. And one of the most spectacular examples of macroevolution paleontologists can brag about. Anyway, it's basically a Rube Goldberg machine in your head. All right, so ear bones can generally tell us two things. Hearing adaptations, which I just spoke about, and you can CT scan ear bones. This is a specimen collected from Onslow Beach. I forget who, though. It may have been Judy Mefford, I think, but I might be wrong about that. But my colleague Travis Park in Australia CT scanned it and ended up with an endocast of the cochlea. And then you can take a bunch of measurements from the cochlea, spit it into this analysis, and end up reconstructing the hearing abilities of your extinct cetacean. So in basal sword whales, for example, if you look at the lower left, tend to hear more like modern baleen whales do. The problem is each new cetacean you add to this data set the whole adaptive landscape of this diagram changes. And it's changed a lot since this diagram was first developed. One of the new fossils, which is a specimen, also Oligocene, from Washington State, was a little dolphin that could not echolocate, so it sat right there in the middle, right next to the Protocetid in red, which is really wild because it either meant that one dolphin lost the ability to echolocate, or even more crazy is that echolocation evolved twice in dolphins, once in the xenoropid dolphins, then once in everything else. So we're still trying to figure that out. We sort of dangerously adopted the scarier of the two hypotheses, which was that echolocation evolved twice, which seems really improbable, but that's evolved echolocation, so maybe not. The other, and this is mostly what I'm going to be talking about for the rest of this, is what we can learn about diversity and taxonomy of extinct cetacean assemblages. We've got a bunch of fossils, some from Charleston, some from Belgrade here, and the upper two rows are all xenorophids. C and D, that's a specimen collected, I think in like 2014 or 2015 by Gary Grimsley. And that is identical to periodics of Albertocetus mephrodorum. On the other hand, E, F, G, and H are close to the brown one on the lower left, which is Echinopsis sanders. And then, of course, there's other ones like the two on the bottom, which don't look like anything from Charleston. And I'll talk about each of these in a little bit more detail. All right, now, if you've got a bunch of fossil teeth and ear bones, what sorts of questions can we ask and answer based upon the assemblage from Belgrade? The easiest one is what cetaceans were there. Ear bones are really diagnostic. There's a lot of different variations between species. And there's a little bit of variation between individuals, but we can generally identify isolated ear bones if they're well preserved, at least to the family level and often to the genus level. And sometimes you may not know which genera you have, but you're like, all right, I know these are all xenorophids because they got feature X, Y, and Z, but there are at least 3 xenoropid genera represented by these ear bones. If you do that, you can address that interesting age that I mentioned. And the age of the Belgrade Formation lends itself towards investigating how rapid or gradual the change from the Oligocene to the Miocene cetacean fauna was. If we look, there are a lot of lineages that are either end right in the late Oligocene or begin right at the early Miocene. Some of these lineages, some of the ranges may need to be updated owing to fossils you guys have found. All right, so let's start. I mentioned that specimen collected by Gary Grimsley. That's on the right. On the left is a periodic associated with the skull of Albertocetus mephrodorum, named after Judy Mefford, and her family in general. And that specimen's from the Ashley Formation. So, and then the one on the right, that's Gary's. He donated that to the Smithsonian after I asked him very nicely. Sure enough, virtually identical. So we've got Albertocetus mephrodorum in both localities. And interestingly, Albertocetus possibly now into the Miocene. So we've got a bunch of xenorophids with a minimum date just extending into the early Miocene, which is these are the youngest xenoropid specimens in the world, which may not say a whole lot because they're basically only known from 2 sites, the Oligocene of North Carolina and the Oligocene of South Carolina. Basically Topsail, Onslow, Belgrade, and then the Charleston area, and virtually nowhere else in the world. Next up, we've got Echinopsis. On the lower left is that specimen collected by Judy Mefford. And then in the middle, that's a periodic that I collected on my very first trip to Belgrade in 2016 that you know, Dave Bohaska was telling me, well, there's a slim chance of finding Oligocene cetaceans, but, you know, we're probably not going to find anything interesting. And we had both kind of decided that we were going to try and find an early dolphin earbone. And I lorded this over him all weekend. So these two specimens of the species indeterminate on the left, that's from Onslow. Mine is from Belgrade. And then a couple more recently donated specimens. And some of these, I can't remember exactly who collected them. Although these ones on the right, I have a strong feeling it was either, I really think Julie Niederkorn donated these, but it could have been Eric or Don Rideout, but I really think it was Julie, sorry. And Sarah's offering to look it up for me. 4277. All right. So the one on the lower right is a very close match for Echovenator, and as are the periodics on the left. The bulla on the upper right is not a real close match for Echovenator or anything else from Charleston. It is a xenoropsid. So if you look, the upper right margin of the bulla has this step-like margin, and that's a primitive condition. Okay, Julie did collect the one in the lower right. Thanks, Julie. More Julie's fossils on the way. Echovenator sandersi is known from a virtually complete skull and pair of jaws and these beautiful ear bones up top. So we've got Echovenator in both the Belgrade and the South Carolina assemblages. Oh, here's one of mine. On the bottom is the biggest xenoropsid bulla I have seen from Belgrade, and that is Xenoropus itself. So unclear if it's Xenoropus sloanii. There's no other species named of Xenoropus yet anyway, but there might be a couple different species preserved in Charleston. So I'm just going to leave it at the genus level, but it's about 20% bigger than the other bullae you just saw. There's also these teeth. And again, I forget who collected them. If I know Eric and Julie are both in on this. So if you recognize a fossil, just interrupt and say that was mine. Just feel free to brag. It's your you're right. 1826. All right. So and here's a tooth and a bulla of Xenoropus sloanii from Charleston. Very close match. Julie collected that tooth also. Thanks, Julie. I know Julie collected this little tiny tooth. If you look, that's a 1 centimeter scale bar. This is really tiny. It is very pretty. This we had on display in the amateur paleontology contributions to science case at the museum for a couple of years. This is a spear-toothed dolphin. So on the right-hand side is Wipatia matafenua, named in Māori from the Oligocene of New Zealand by my PhD advisor. We have these spear-toothed dolphins from the Oligocene of Charleston as well, and now South Carolina. And there's a few others known from, I think, Eastern Europe and possibly Malta. So they were likely worldwide, unlike the xenorophids, which seem to really be homegrown Carolinian fossils. Most of our Wipatiids from the Chandler Bridge Formation are a bit bigger. So this is to the same scale. And you know, the number of cusps doesn't match up. It's got fewer cusps, it's got more rugose enamel. It's quite a bit bigger. The roots are fatter, etc. But there is one jaw from the Ashley Formation over at Charleston Museum that has really tiny teeth like this. It's not a perfect match, but regardless, it's probably the same family, possibly an unidentified genus or species. And there's probably a half dozen Wipatiids known from the Oligocene of Charleston. And not all of them are known from teeth, though not all are as small as the specimen on the upper right. Some folks have found fossils of Ankylorhiza. So we've got a tooth on the lower left that was collected by Julie also. And then above that, right next to the 1 centimeter scale bar, was recently donated by Elizabeth Smalley. And this bulla on the right-hand side, all of these are specimens of Ankylorhiza, our giant scary dolphin from the Oligocene of Charleston. The skeleton that we published on last year, CCNHM 103, that's the tooth of it, and it's nearly identical to the tooth that Elizabeth Smalley donated. With the exception, it doesn't have any of those little bead-like cusps on the left side of the tooth, but those are only present on like the last two positions. So I think the similarities are enough to identify it to that species, Ankylorhiza tiedemannii. There's this bulla on the lower right. It's a bit smaller. We've got two species of Ankylorhiza. Unfortunately, tiedemannii doesn't have any ear bones preserved. There's a larger species that has different looking teeth, and it has ear bones. But the ear bones are a lot bigger than the ear bones of Ankylorhiza from Belgrade. So it's possible that the bulla also represents tiedemannii, which is a smaller species. The new unnamed species of Ankylorhiza is about 20% bigger. It's got a 1.25 meter long skull. So it's a bit bigger even than our scary dolphin we named. That's another future project of mine, but so far we only have evidence of tiedemannii. So the bulla may represent tiedemannii also, or something else. Some unnamed third species of Ankylorhiza. So we're gonna just call that Ankylorhiza species indeterminate. And the last of the Oligocene cetaceans from Belgrade, this is a big bulla collected by Eric Sadorf of an eomystacetid baleen whale. Eomystacetid ear bones look a lot like really huge dolphin ear bones. They don't look much at all like Miocene or modern baleen whale ear bones. They've still got a big crease in the back, and they're quite wide and kind of triangular. This is pretty close to Eomystacetid whitmeri, and it's a lot larger than that Ankylorhiza bulla. So it's definitely not a big dolphin because it's just way too big. It is a pretty good match for things like Eomystacetid whitmeri, though. This is the bulla that goes with that same skull I already showed. It's slightly bigger. It could represent something like Micromystacetid, which is known from the Ashley Formation, or some sort of unnamed Eomystacetid. Eomystacetids are known from Australia, New Zealand, Japan, Washington, Oregon, as well as Eastern Europe, and obviously the Carolinas. So it's possible it's an Eomystacetid that we don't know of from Charleston. Now we start finding some fossils, or we get into fossils that are a little more similar to what you'd find at Lee Creek Mine if you're looking through the Pungo. Or at Calvert Cliffs for that matter. So these are little ear bones of a Urina delphinid, which I call swordfish dolphins. These are the extremely long snouted dolphins that are very commonly found at Calvert Cliffs. I think they're the most common cetaceans. The lower jaw doesn't extend to the tip of the upper jaw, and then the upper teeth end there. So the front half of the upper jaw is toothless. It's kind of like a police baton. I'm not really too sure what it was used for. Actually, it's more like a pool cue, and literally about the size and shape. So these ear bones, these are pretty close matches to ear bones of Xiphiacetus or possibly Schizodelphus from the Punga River limestone at the Lee Creek Mine. And they're tiny. These are the smallest ear bones we have. There's also a periodic collected, I think also by Julie, though I screwed up and forgot to get a photo of it. In addition to Ankylorhiza, we have fossils of true squalodon. So on the bottom is a really large periodic. This is actually figured in the North Carolina Fossil Club marine mammal book. Eric Sadorf collected this, and this appears to represent a really big species of squalodon. It's not Squalodon calvertensis. Squalodon calvertensis is a little bit smaller and not as robust. We unfortunately don't know what the periodic of the giant species Squalodon whitmeri looks like. But this could be Squalodon whitmeri. It could also be an earlier species, given that whitmeri and calvertensis are from the late early Miocene and middle Miocene, rather than earliest Miocene. It could even be something like Squalodon berriensis from the early Miocene of France. And then on the upper left is a tooth of Squalodon or possibly Eosqualodon. I'm leaning more towards Squalodon these days that Don Rideout collected. And it was actually the first specimen from Belgrade donated to our museum. So thanks to Don for that. Some of the teeth of Squalodon are really distinctive in having serrated cutting edges, like a great white shark, which is kind of scary to think about, a dolphin with serrated teeth. Ankylorhiza does have some serrated teeth, but they're generally all the way in the back of the jaw, not on these. This is like a anterior cheek tooth or maybe a posterior incisor or canine. So it's from the front third of the snout. Ankylorhiza doesn't have any serrations in those teeth, and its teeth are also a lot larger and beefier. We also get squalodelphinids. These are some of the more common ear bones found in the Punga River limestone. I think these were collected by Julie and Eric, if memory serves. And they're very similar to ear bones identified in the Lee Creek Mine 4 volume by Whitmore and Kaltenbach as Phocogeneus, which was named, I think, by Cope based on a single tooth back in the 19th century. And Remington Kellogg referred a partial skeleton, including vertebrae, teeth, and ear bones. No skull fragments, but ear bone bits. And then more material was found at the Punga River mine. Punga River limestone in the Lee Creek mine. These are very close to specimens from the Lee Creek mine. Here's a specimen from the McDaniel collection. So the complete specimen from Belgrade has a rounded anterior process. That's the thing sticking off to the left on the upper picture of that complete periodic. And it's a lot pointier in the specimen from the mine, but there are other specimens from Belgrade that have a pointier anterior process. So maybe these are con-specific or at least con-generic. The real clincher for me, though, is the bulla, which is surprising because the bullae are usually less diagnostic than the periodics. The bulla has, if you look at the margin on the bottom, it's got this double hump. And that is unique to specimens of Meticinia / Phocogeneus. The genus level taxonomy is in flux right now. We're waiting on some of my European colleagues to publish on a skull from France and restudy a skull from the Lee Creek mine. It used to be called Phocogeneus. It's possible it's this thing from France called Meticinia. So I'm just saying it's both for the time being. But among squalodelphinids, which are relatives of long-snouted platanistid dolphins, including the modern Ganges River dolphin in India and Pakistan, this double-humped margin that you see on the bottom there is unique to this taxon. So I'm pretty sure all of these represent the same genus and possibly the same species. And the Belgrade form is a little bit smaller and possibly ancestral to the form you get in the Pungo. certainly congeneric. Whichever genus it is, it's the same one. One of the surprising specimens is this one. I can't remember exactly who found this. I have a feeling this one was Julie as well. This is a more recent find, and when I first saw it, I thought this has got to be a Pliocene common dolphin from the Duplin Formation. You know, you do find occasional great white shark teeth and megalodon teeth at Belgrade. And this, I thought, nah, this is way too advanced looking to be an Oligocene specimen, or even early Miocene. But it's got this weird little, if you look right above the scale bar at the top picture, it's got this like kink in the bottom margin there. And that big thick part, that's the involucrum, the thickest part of the bulla. There's not a whole lot of dolphins that have a constriction in that part. And there are all of those that do have it, though, are Kentriodonid dolphins. Kentriodonids are probably the group that oceanic dolphins like killer whales, bottlenose, spinner dolphins, so that's the oceanic dolphin group, the delphinids, true porpoises like harbor porpoises, the vaquita, Burmeister's porpoise, etc., as well as the beluga and narwhal. All three of those modern families probably evolved from Kentriodonid ancestors during the Middle Miocene. If this is the Kentriodonid, and I'm pretty sure it is, this would be one of the earliest known Kentriodonids in the world. There are possible Kentriodonids from rocks that are slightly younger from New Zealand that some of my lab mates were working on when I was still at University of Otago in New Zealand. So that's kind of a surprising one. We also have a possible sperm whale tooth, and it's real crappy, and it's possibly a busted up crocodilian tooth, but it does have several internal growth bands that look enough like a sperm whale. And it's got this little collar in the enamel, which you generally only see in mammals. You don't get them in crocodilians, and it's this little bulge. And if you look in the picture on the right-hand side, looking straight down the axis of the tooth, you see that dark band, that dark shadow. That's actually where the enamel's been broken off the crown. And there's this little white band around it with another dark band below it. That's actually a little bulge in the enamel that forms a little ring, which you see in a lot of dolphin teeth. And we think enamel growth gets all screwy when the animal is weaned. So it's actually been targeted for isotopes, looking for isotopic changes in growth and trying to see, see if we can learn anything about the change in the animal's physiology during weaning. Anyway, hopefully there will be better sperm whale teeth found there in the future. But sperm whales show up for the first time during the earliest Miocene. There is a purported Oligocene sperm whale from Kazakhstan named Pherecetotherium kelloggi, originally interpreted as a toothed baleen whale, but more recently reinterpreted as an early sperm whale, though it really does need to be redescribed anyway. So this would be one of the oldest records of a sperm whale in North America. Unless it's a croc tooth, in which case, not very interesting. This is one I found, so I'm not ripping on somebody's donation here. All right, so that's it. So there's a few other. I didn't include all the specimens because I didn't want to eat up too much of your time. But those are all a lot of the really good ones. So what answers do these fossils have for us? Well, about half and half of the dolphins are either from the Oligocene or the early Miocene. And there's one rare example of an Oligocene mysticete. So we've got the Oligocene taxa there in the blue stars. So already fossils you guys have found extend some of those bars quite a bit. And then there's a bunch of early Miocene dolphins as well. No early Miocene baleen whales. And I mean, taxa usually thought to be representative of the Oligocene or the early Miocene. Obviously, these cetaceans are all from roughly about the boundary itself. Unless these fossils are reworked, and it's possible some of the Oligocene taxa might be reworked from the River Bend Formation. Unless they all are, this would suggest a more gradual change in faunas across the Oligocene-Miocene boundary than we would have known otherwise. And with that, I'm going to open it up for questions. So, but before I do, thanks to you guys for inviting me and donating a bunch of great fossils to our collection. They are modest in size and occasionally preservation, but highly informative. And you can see there's a lot you can do with random ear bones you find out on the ground. So in particular, I'd like to thank Gary Grimsley, Trish Kohler, Julie Niederkorn, Don Rideout, Eric Sadorf, and Dave Sanderson for donating all these. And thanks to Dave Bohaska and Jonathan Geisler for letting me talk to your ears off about these. So anyway, thanks again. And if there are any questions, I'd happily take them.
Well, thanks for that, Bobby. That was fascinating. I can't wait to bring you the box of ear bones I have from different sites so that you can take a look at them. You've given me a real new appreciation for how interesting they are. So does anybody in the membership have any questions? You can just go ahead and unmute yourself, please, or write it in the chat if you want, whichever one you're most comfortable with.
First off, Bobby, I want to give you my apologies for donating my stuff from the River Bend to the Smithsonian instead of you. I wasn't really aware that you were studying that when I gave them all to Dave. So I'm sorry you can't get up there to see them because it's too bad.
That's all right. I've gotten some photographs of Smithsonian specimens already.
Yeah, it's those two brain endocasts, and then there was a couple of pretty primitive whale teeth, all from Topsail.
Okay.
So
yeah, I thought I remember your names on. I could visualize the specimens right now.
Yeah, and I have photos too if you're interested in them, but they wouldn't be probably the quality that you'd get from the Smithsonian.
Well, I'll two comments there. I have been discouraged by Nick Pyenson from publishing on those specimens. And I'm not going to go into the politics of why, but he has not exactly made any leeway on that. He just doesn't like me very much. Anyway, so that's another reason why I'm restricting this current study to fossils from Belgrade Quarry itself. There's also there's a couple skulls that are as yet undescribed from Topsail or Onslow rather. Sorry. So somebody else can come along and do the Onslow and Topsail descriptions.
Got it. Are you familiar with Clark's Quarry?
I am not.
Okay. Somebody else in the group here will have to brief both Bobby and me on the specific age of the Clark stuff. I know that there's the there's an upper Eocene layer that I get a lot of my crystallized invertebrates from, which is I'm going to try and think of the name of the formation because I'm blanking out now because I'm thinking about the river bend. So it's knocked the other thing out of my head. Anyway.
Silverdale?
Nope.
Okay.
No, it's like the what?
Dawn, was that you? Could you repeat that?
Spring
The
Garden.
ESC,
Spring Garden member, right?
it's the Spring Garden member of the Castle.
Spring Garden member of the
Correct.
Castle Hane. Okay, so
Yes.
Spring Garden member of the Castle Hane, and then there's some
Mhm.
younger overburden as well. Well, from said quarry, I did find this.
Oh, that's a beauty. That's
Want it?
that'll actually be a Pliocene specimen. That's Balanoptera borealina by the looks of it,
Okay.
which is well known from the Yorktown.
Okay, then it's
Yeah.
not anything you want. And you're welcome to it if you want it. I don't do vertebrates. I just have them if I find them. Okay, just thought I'd show it to you in case you wanted it.
Yes, Bobby, the Clark's Quarry has Castle Hane, Spring Garden member of the Castle Hane, River Bend Formation, and then it has a Yorktown layer, as well as a pretty good Pleistocene layer on top of it. So it's a very interesting quarry.
Is this the one that Bohasco was telling me about, some quarry, and maybe it's close up to the northern border of North Carolina?
No,
No,
this
that's
quarry is
Rocky
very
Point.
close.
Okay. Oh, that's right.
Yeah. Rocky
Yeah.
Mount is the one. It's the Hanson Quarry
Yeah.
up by Rocky Mount. This one also is known as the Newbern Quarry.
Yes. Mhm.
Oh, okay. Yes, I've heard of that one.
Yes. Mhm.
All right, are there any other questions for Bobby?
Awesome
Okay, well,
talk.
with that meeting now, I want to thank everybody for taking the time to be here and for all your interactions with Bobby. First of all, I'd like to know if there's anybody on the line today on the meeting who's here for the first time, is a first time joiner or visitor to the North Carolina Fossil Club. All right, I don't see anybody jumping up and down on their videos there. So, all right, we'll go on. The other announcement is just another sad. Oh, Joyce D, first time meeting. Joyce D, you want to unmute yourself and tell us a little bit about yourself? You don't have to. You can just say no.
Well, a lot of you guys already know me. I've been on some of the club trips and have been a member for a couple of years. Was a volunteer for the Aurora Fossil Museum for just over a decade and has been collecting for a long time. So a lot of you guys already know me. And congrats, Julie, for all the stuff that you donated. Yeah, it was awesome.
Yeah, absolutely. Nice for our members to get a little bit of recognition. Well, welcome to your first meeting, and we will look forward to seeing you in real life sometime in hopefully the not too distant future. The next little bit of news we have is another bit of sad news. Unfortunately, Becky Heine, who many of those of you who have been longtime members of the North Carolina Fossil Club will remember. She was one of the bus drivers into into the PCS and mine when it was open, and she was just one of the most lovely, generous people. And she's going to be missed. She passed on this last month, so another one of our members leaving far too soon. We can all now move on, please, to reports of officers and committee, and I'll ask Terry to read the minutes and see if there are any corrections. Terry, please.
Yes. All right, this was from January 17th. The meeting was opened by Ramona at 1:30 p.m. The program was dinosaur paleopathology, what dinosaurs sick and What Does That Tell Us by Dr. Andrew Heckert from Appalachian State University. Announcements: Charlie Causey has been appointed copyright consultant to assist the board, and specifically Joy Harrington in the management of the club's copyrights. It was reported 2 longtime members, Tom Burns and Jerry Case, passed away. Trish Kohler has volunteered to become cheer coordinator for the club to acknowledge important events for members. Reports of officers and committees. The minutes from the November meeting were submitted by Terry Denny. There were no new corrections. Minutes were approved. Treasurer's report: David Sanderson reported the current balance was $24,560.39, and memberships can be renewed through PayPal on the website or mailed into the PO box. Website membership: Mike Bruff reported there were 103 memberships, 46 individual, 13 honorary, 44 household. 74 memberships were renewal, 16 were new. Books, etc. Gary Lewis reported sales are brisk. Some delayed shipments due to holiday postal service. 2021 calendars sold out. We're down to 1 copy of volume 1 and 2 copies of volume 4 part 2. Gary will work on arrangements to reprint and discuss with the board. The newsletter: Eric reported the next Janus was March 1st, 2021. Articles due 1 week before. Ramona has encouraged members to share stories for the next issue. Outreach: Daryl Grader was not present. Terry Denny shared about her virtual outreach for 2nd graders at Leadmine and at Lacey Elementary Schools, and a STEM night at Lacey. And she has 3 more upcoming presentations. Grants Committee: Linda McCall reported for Don Rideout that the committee will be meeting soon to finalize a proposal for the board. New business, board notes: Ramona reported the board had met and voted to plan for an in-person fossil fair in November, hoping that it will be possible to have live events by then. She will be making contact with prospective hosts for the event. The club will need a volunteer to lead this event once location is settled. Appalachian State was mentioned, but it will not be possible this year due to scheduling conflicts. And out-of-state locations were mentioned, but it will need to be investigated. And it will also be subject to whether or not it will be safe to hold an event and what COVID numbers are at that time. The board also decided to send virtual copies of Janus to certain Corey members and others that provide opportunities for the club. Field trip update: Linda McCall reported Corey's are still on hold due to COVID restrictions. There may be dates in spring if there's a drop in positivity rates. Outreach and events upcoming: Daryl Grader was not present. Ramona shared Shield Museum's having a fossil fair event scheduled out over several days in February to allow social distancing, looking for exhibitors. Meeting adjourned at 3:20 p.m. The next meeting, March 21st, 2021 at 1:30 p.m.
So are there any corrections to those minutes? Could I have a motion to the floor that they be accepted as read, approved and accepted as read?
I'll make a motion to approve the
That-
minutes.
Thank you, Linda. May I have a second to that
Second.
motion? Fantastic. Are there any objections? Then we will approve those minutes as read by unanimous consent. All right, Mike, is there anything you want to say about the membership or about the website?
Well, we have the treasurer's report first.
You're absolutely right. Oh my goodness. David, could you please give us the treasurer's report?
Okay, the current balance is $26,000. I mean, $26,022.45. Got a healthy balance. Do you want me to do a quick recap on the year-end treasuries report? That was published in the Janus, but I'll do a quick recap. We started 2020 with $22,629. For income, we had a little over $4,000 in dues, about $5,700 in book sales, calendar sales, which was a total of about $9,800 in income. If you back out $105 for PayPal fee, we had about $9,700 in income. For expenses, very minimal this year. Or last year, mainly due to our virtual meetings. We only had $200 in club meeting expenses for picnics, things of that source. We had a $1,000 donation to Emily Simpson, and we spent about $4,400 in printing of books and things of that sort. So we had about $8,000 in expenses. So started the year with 22,600 and ended up with 24,410. So we got again, we have a very healthy balance in our report. So any questions?
All right. Well, thank you, David. Diane, did you have a question? I saw you unmute yourself now. All right. Then I'll go on and refer back to Mike and ask about any updates for the membership or the website.
Well, good afternoon. No updates on the website or anything like that. As far as the membership report, there's a total of 144 memberships, which is up from January, probably approximately 40, 67 households, 64 individuals, and 13 honorary members. That's it.
All right, Mike, thanks very much for that. I'll then go and turn it over to Gary and ask that you just give us a quick update on our publication activities.
Book sales continue to be about normal. Probably average about one, maybe two orders a week. Right now, we are completely out of volume 1 books, and we are completely out of volume 4 part 2 books. And I am working with Richard Chandler to get updated files so that we get those off to the printer and get more printed. So we'll get those back in stock. Beyond that, everything's going well.
All right, that's fantastic. Thanks very much. Move on then to the newsletter. Eric?
The next newsletter will be out around June 1st. So if you want to submit something, get it to me about a week before then.
And I'm going to do my little plea and beg that that the membership share what they've been doing for the last year. I mean, this has been a unique experience for all of us. I spent a lot of time curating. I even got really desperate and looked at fossil auction sites at one point. So seriously, anything that you've done that you can kind of share the joy, we would love reading about it in the next issue of Janus. You don't need to be professional writers, and it doesn't need to be more than 2 or 3 or 4 sentences. And you don't need to give away any secrets about your favorite sites. Just kind of tell us what you've done to keep the joy alive. All right, now we will move on to outreach. Daryl, I did see you on the, yes, I see you there. Is there anything that you can add to that for us? Daryl, we can't hear you. I can see that you're unmuted, but. Yeah, Daryl, we're still not hearing you. All right,
I can share that I did 5 outreaches since the last time we met, and I have a couple more pending if the dates can be worked out. So I'm starting 4th grade now. So I've finished all the 2nd grades.
That's outstanding. Anybody else have anything they want to add?
Yeah. I was asked by Davis Drive Elementary to put together a video on fossils. So I've just completed that, and I'll be sending it off to them.
Oh, how cool.
So I put that together. I also worked the Dino Fest recently. So if any of you went, you may have seen me with a baby dinosaur on my arm. arm at the end, or working some of the others. I did tell some of the people that I was a paleontologist, and it thrilled them. But the biggest thing was seeing the looks on the faces of the kids. And as much as possible, I did try to spread the word about the Fossil Club. So if people showed interest, I did mention the Fossil Club and told them to look it up. Just look up NC Fossil Club on the web and it should pop right up so they could get information about the Fossil Club.
That's great. Thank you. Anyone else been doing anything in their communities? All right. Well, I think that opportunities for outreach are going to be increasing over the next several months. I think talking about schools going back, and I'm sure that they will hopefully be looking for people. Although, you know, there may be a whole new set of rules. Who knows? We'll keep posted on that. All right. Does Dawn had to leave? Was there anyone who had anything that they could say about the grants committee? I know, Linda, you're on the committee. Do you know if there's been a subsequent meeting?
Jim and I are both on the committee, and we have not had a subsequent meeting. So as far as I know, there's no more update than what I gave you the last time. I talked to Don yesterday, and he said that he had been really, really busy, that he wanted to get together with the rest of the committee and sort of finalize what we had going on, but we just haven't had a chance to do that yet. So hopefully that will happen sooner rather than later, and we will have an update for you shortly.
Fantastic. Do you want to stay online then to do a field trip update?
Sure. It's under new business, but oh yeah, I'm next anyway. So I talked to Danny about a week ago. He's our outreach person from Martin Marietta, and they are looking at possibly starting up the end of May, early June is the word that he's getting. So we haven't had that concrete yet. But that's what he was saying that they were hoping for. So be watching your inboxes, etc. for, you know, later on, the end of spring, early summer, we might be able to get back into the quarries, provided everything continues along the path that it currently is. I am currently considering a trip to Old Dock, although it's really, really small now. So that would be a really, really small trip for really, really few people. But I have talked to the quarry owner, and he is amenable. So that may be happening. And I think Rick is here, and Rick was going to talk a little bit about the trips that he leads and if he had any dates in mind. Is Rick still here? Nope. You see Rick anywhere? Rick Trone?
I see his name.
Rick is
no
on and unmuted, but we're not hearing anything from him.
Yeah, all right. Just another technical glitch. All right, well, that takes us to the last item, and that's talking about the North Carolina Fossil Fair. As mentioned at the last meeting, I was going to reach out with some of our partner museums, and so far nobody is planning events. So we will continue to reach out and try and find someone who would be happy to partner with us to try and plan an event for the fall, given all the uncertainties we have. So that's kind of where that is. I'm still hoping somebody will step forward in spite of all the uncertainty and agree to be the leadership, take a leadership role in managing the fossil fair for this fall. So please, 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 if you are interested in doing that, let me know by any means possible. Oh, Rick is in chat right now. He says that he may plan a Greens Mill run trip starting in April and moving forward. Fantastic. Technology. All right. Well, is there anything anybody else in the membership wants to, oh, I will also take, don't leave chat yet. I will also take people to the Tar River and Swift Creek but those are dependent on river levels, which are unusually high this time of year. Indeed, they are. All right, is there anything else under new business that any member would like to bring up for consideration?
I'm just gonna say, guys, keep your eyes on your email for things from Rick or from me about any pending trips coming up.
And Deborah Palmer is asking, will members be able to access the demonstration recording? I'm going to interpret that to mean that you can access Bobby's talk. Yeah, okay, great.
Ramona,
Yes,
you can hear me?
yes,
Finally?
I can.
I think I figured out what was wrong. One further update on the outreach. I did want to say that the Shield Museum did have their fossil fair. It was a different type of fair, but from everything I heard, it went very well. They had a good number of people there. And I'm happy to report that Ruff and Tucker was one of the people who actually displayed at the fair for them.
Congratulations, Daryl. Thank you. I know how exhilarating it can be when you get your microphone to finally work. Well, thanks for that. Yeah, MC Douglas out at Teal is one of the people that I'm waiting to hear whether or not she'd, kind of take a walk on the wild side with us to try and plan something, given all the uncertainties. And, they pulled it off, and she has seemed quite willing to adapt. So yeah, maybe we could, she'd be happy to work with us for this fall. We'll see.
Very good.
Okay.
Mike says that he'll send out a link to the recording in chat.
Thank you, Linda, for keeping me honest. Appreciate that, Miss Vice President. All right. Anything else from any of the members?